Okay, ladies and gentlemen, you are welcome. So in this video, we are going to look at the laws of inheritance. This law of inheritance, they are the laws that are trying to explain how traits or how characters are passed from parent to offspring. So please don't forget to subscribe and join my YouTube community. So introduction to laws of inheritance. So the laws of inheritance explain how traits are passed from parents to offspring, and these laws are fundamental to understanding genetics and heredity. So if you are able to understand these laws, it will actually give you a good understanding of how genetics and heredity are. So Gregor Mendel is considered as a father of modern theory of inheritance. For in fact, he was also considered as a father of genetics. So these are the following laws that we are going to look at in the present video: the laws of segregation, the laws of independent assortment, and then the laws of dominance. So these are the three things that we are going to look at in this video. So now, starting with the Mendel's first law of segregation, or we call it Mendel's first law of inheritance, or you can call it law of segregation. So according to him, according to Gregor Mendel, the law states that each individual has two alleles for a trait. So it means that each trait, the color of the skin, our shortness, our tallness, intelligence are all examples of traits. So every individual or each individual has two alleles for a trait. And that two alleles, it can be homozygous or heterozygous. What is homozygous? When we said homozygous, it can be capital X, capital X. This is homozygous. It can be heterozygous. Heterozygous, it can be capital X, capital Y. Or it can be capital letter X, small letter X. This is heterozygous. So every trait in an individual has two alleles for that trait. And these traits, this allele usually separate during gamete formation. Like for example, let's say we have X and Y. So during gamete formation, these alleles will be separated. That is, we'll have allele X and allele Y during gamete formation. So offspring inherits one allele from each parent. So that is what usually happens. Like for example, first we have two alleles for each trait, and these traits, this allele will be separated during gametes, and each of these alleles can be inherited from a parent, resulting in genetic variation. Like for example, let's say from the mother we have, let's say, capital X, capital Y, and from the father we have small x, small y. So according to this law of inheritance, these allele will be segregated or will be separated during gamete formation. X will be separated, Y will be separated, and then small x and small y will all be segregated. So after this, each of the allele can be inherited to the offspring from the mother. So therefore, that is why you can have a different crossing. Let me delete this so that we can see more of this demonstration. So I think you understand. First, each individual has two alleles, and this allele will be separated during gamete formation. An offspring will inherit one allele from each parent, which resulting in genetic variation. So this segregation of alleles occurs randomly. So it occurs randomly, and this random occurrence of this allele ensured diversity in traits. So that is why a child cannot be exactly as his father. It cannot be exactly as his mother. Rather, you will have a hybrid child that have a trait from the mother and a trait from the father, because each allele is inherited from either a father or a mother, which result in the formation of a two allele. And those two alleles in the offspring are coming from the father and the mother. So let's look at it. So according to this Mendel's law, you see we have this inheritance. This is AA. We have AA. You see, this is capital letter E and small letter AA. So you see, they are now segregated. They are now separated. As we can see, they are now separated. So after separation, then they will now combine. You see, let's say this is a father and this is a mother. So as we can see from here, the allele from the father for this trait, they are segregated. So you see capital A here cross with small letter a. That is why we have capital A, small a. This capital A again also cross with this small letter a. That is why we have this. This one also cross with this. We have this. This one with this, we also have capital A, small a. So that is actually the law of segregation of gene. So the first law of Mendel is also known as the law of uniformity. It established when two individuals homozygous for opposing traits are crossed, the offspring of the first generation, that is F1, is called first filial generation, will be heterozygous and will only show the phenotype of the dominant parent. So as we can see, if this allele, you see this is capital A, small a, small a. So the capital A here signifies a dominant character, and small a signifies a recessive character. So remember, a dominant character is a character that masks the effect of the recessive character. So that is why, let's say this capital A is a dominant character for red color. So it means that after you cross all, this is our F1 generation, that is first filial generation. So as you can see from this first filial generation, all the offspring here are heterozygous. Why is it that it is heterozygous? Is because capital A, small a, capital A, small a. But this one is heterozygous recessive because they are all small a, so they are the same. So this is homozygous recessive, and this is homozygous dominant because they are all capital A, capital A. So at the end, because A are dominant character, so and it's responsible for controlling a red color, and small a, let's say, is responsible for controlling black color. So because A is a dominant character, so it masks the effect of small a, which is the black color. So that is why all the offspring will show the same phenotype. So it will be heterozygous and will only show the phenotype of the dominant parent. So here the dominant parent, let's say, is the father with AA. So it means that all the F1 generation are going to be red in color. So all descendants will share similar characteristics, demonstrating uniformity in phenotype. So it means that all the phenotypes are the same. So phenotypically, they are all red in color if you consider capital E as a dominant character. So this law lays the groundwork for understanding genetic inheritance and allele segregation, providing a crucial foundation for classical and modern genetics. So you can see capital A, capital A, small letter A. So the first segregated, this is the segregation, they separated during gamete formation. And after the segregation, then they cross with one another. Then we all have the same heterozygous allele, which express the same phenotype. So now, the law of independent assortment, that is the second law of inheritance, it is a law that this law states that the inheritance of one trait does not influence the inheritance of another trait. So in the case of law of independent assortment, it means that the inheritance, if an individual inherits one gene, it doesn't mean that it will influence the inheritance of another gene. So the genes for different traits are inherited independently of each other. So that is what the second law of Mendel is trying to demonstrate. So if you have two different traits and one of the traits are inherited, so it doesn't mean that it will have effects on the inheritance of another. So because they are genes of different traits, so since they are the genes of different traits, so they are going to be inherited independently of each other. So that is why it is called the independent assortment. So this law explains the genetics variation observed in offspring of multiple traits. So now let's look at the demonstration here. We have here the Mendel's second law of inheritance, which is the law of independent assortment. Here we have parent generation. We have round and yellow. Then we have wrinkle and green. So if you look at this, these are two traits. Round indicates the shape. Yellow is color. Wrinkle is also a shape, while green is also a color. So if you look at it, we have capital R, and capital Y, capital Y. So capital R is responsible for the round, while capital Y is responsible for yellow. And then, of course, the wrinkle is small letter RR, and then the green is small letter YY. So what happened? So these genes will be first assorted. How is it going to be assorted? Capital R will be assorted. Small r will also be separated during gamete formation. And then the next one is capital Y will be assorted. And then small r will also be assorted. So after all these are assorted, what happened? So we assorted capital R, then we assorted small r, and then we assorted capital R, and then the next one we assorted small y. That is what I want to say, not small r, small y. So what happened next? This capital R will cross with capital Y, and this will give us capital R, capital Y. And then at the same time, this capital R will cross with capital R, small y, and then at the same time, capital R, sorry, small r with capital Y, we have small r, capital Y. And then the small r again will cross with small y, then we have small r, small y. So if you look at this, In the F2 generation, we are now going to take this. So look at this here. We have capital R, capital R, capital Y, capital R, small y. It means that we take the sum of this like this if you look at it very clear. And then we also have the same thing here vertically. So then we now demonstrate all this in a Punnett square. So this is our Punnett square. So we now cross this with this, and that's why we have capital R. You see capital R, it will cross. We have capital R, capital Y. Then R, capital Y will cross with this. So we have, it will cross with this. So that's why we have capital R, capital Y, small I. That is why we have small y. That is why we have this. Capital R, capital Y will cross with this. So that is why we have capital R, small r, capital Y, capital R. And then this also again would cross with this. That's why we have capital R, capital Y, small y. And then this one also will cross with this again. So that's why we have capital R, capital Y, small y. And then this one again will now cross with this. So that's how it follows. So we do with this. Then the next one is this one with this. Sorry, it will start with this, then cross with this, cross with this, cross with this, and cross with this. That is why we have all this. So in the second fillet generation, we have the following characters. So we have round and yellow. So round and yellow, why is that it is round and yellow? It's because if you look all this, there is capital R and capital Y in them. And the small y and the small r are all recessive. That is why we have round and yellow. And then, of course, for round and green, we have this. So that is why at the end, if you look at it very well, the issue or the concept of recessive and dominant characters also plays a role here. So after F2, this is our F1 generation. This is our F1 generation. And this is our F2 generation. So for the F2 generation in the law of independent assortment, look at the ratio. We have 9:3 is to 1 is to 3 is to 3 again, then is to 1. Let me delete this so that I can demonstrate it very well. As we can see from here, we have 9 ratio 3 ratio 3 again ratio 1. So this is the law of independent assortment. So the next thing is dominant and recessive traits. That is what we are going to look at. So a dominant is a character that expresses when at least one dominant allele is present. So we have recessive and dominant allele. So whenever we have a dominant allele, it always expresses. It always expresses. While a recessive trait is a trait that only expresses when two recessive alleles are present. So if you have two recessive alleles and they cross with one another, so they will express. But if there is a recessive and there is a dominant, recessive doesn't express. It will be masked. So therefore, it's a dominant allele that will be expressed. So dominant allele are expressed when at least one dominant allele is there. But for the recessive, the two recessive alleles must be there. But we also have codominant trait. A codominant trait is a trait that has equal effect or equal chance of being expressed, like in the case of blood group AB. So the combination of alleles determine the phenotype of an organism. So when alleles are combined together, that is what exactly determines the phenotype of an organism. Like for example, look at it. We have capital R as a dominant allele and small r as a recessive. So if you now have, let's say, of course, remember each organism has two alleles. So we have capital R, and we have small r, small r. So if you now cross this, the red means, the capital R means red flower. And then the small r means pink flower. So what will happen? So if we now cross this, let's say we have this one cross with this, we have capital R, small r. This one cross with this capital R, small r. This one with this, we have capital R, small r. And this one is capital R, small r. So if you look at it here, ladies and gentlemen, all this is the F1 generation. So from this F1 generation, all of them phenotypically, they are red in color. But if you have, let's say, capital R, small r, capital R, small r cross, we have capital R, capital R. This one with this, we have capital R, small r. This one with this, capital R, small r. This one with this, then small r, small r. It means that we have 3 red flower and 1 pink, because R and small r, they are recessive. So you see, recessive trait only expressed when there are 2 recessive allele. But for the dominant allele, if at least there is 1 dominant trait or dominant allele, then it will mask the effect of the order. So that is dominant and recessive. So the next thing is Punnett squares and predicting inheritance. So Punnett squares are diagrams that are used to predict the probability of offspring inheriting particular traits. And then they help to visualize the possible combination of parental alleles. And these tools are essential for understanding genetic inheritance patterns. So you see, these are the importance of Punnett squares. It is a diagram that used to predict the probability of offspring inheriting particular traits. Like, for example, if you have a father and a mother, and with Punnett square, you can predict the inheriting patterns of the offspring. And it also helps to visualize the possible combination of the parental alleles. And it's also help in understanding genetic inheritance patterns. So this is a Punnett square, as we can see. So we have capital AA and small aa. So after the generation, 200% of the offspring are AA dominant, because A will all dominate. But of course, for the tats, so you can decide to use Punnett square like this, or you can use any I think mostly the most important thing is you can use the Punnett square. So this is a typical example of Punnett square. So now, modern extension and application. Advances in genetics have expanded our understanding of inheritance beyond Mendel's law. Concepts like incomplete dominance, codominance, and phylogenetic inheritance explain more complex traits. So understanding inheritance laws help in fields like medicine, agriculture, and genetic counseling. And as we can see, the real life application of modern Mendelian genetics, it helps in understanding evolution, it helps in understanding pharmacogenomics, biotechnology, biology conservation, and forensic sciences. So ladies and gentlemen, without wasting much of our time, this is where we are going to stop, and in the next video, we are going to look at the concept of incomplete dominance, codominance, and polygenic inheritance. Thank you very much, and see you in my next video.